The primary statutory activity of the Institute of Human Genetics of the Polish Academy of Sciences is basic research in medical sciences. The Institute has significant accomplishments in the development and introduction of modern diagnostic methods, innovative drugs, and therapeutic approaches. For years, we have conducted research on genetic diseases while paying attention to their diagnostic aspects.
We were the first in Poland to start the research on the causes of keratoconus, a disorder of the eye characterized by the conical shape and thinning of a cornea. Currently, we are looking for the mechanism of the formation of a characteristic conical shape of the cornea and changes in the corneal layers. Working with ophthalmologists, we disseminate knowledge about keratoconus, which significantly increases the awareness of the medical community and the recognition of the disease in Poland.
Late diagnosis of head and neck cancer is a major problem in laryngeal oncology. We therefore focus our research on designing a non-invasive genetic test based on the analysis of peripheral blood, intended to speed up therapy and indicate patients with high risk of recurrence. For example, tumors of salivary glands are a significant problem. To better understand their causes, experimental models that could be used by scientists worldwide are necessary. To meet such needs, we have established a unique, patent-protected cell line model of malignant salivary gland tumor.
Genetic diseases of the respiratory tract are one of the main research areas of the Institute. We have co-created the first in Poland model system of molecular diagnostics of cystic fibrosis. We also study genetic basis of PCD, another pulmonary disease caused by the impaired movement of respiratory cilia. We are co-authors of European guidelines for the diagnostics of these diseases, and we have initiated formation of Polish PCD Society. We participated in the development of an innovative inhalatory drug for viral infections, and we have demonstrated that Fryderyk Chopin, the greatest Polish composer, died not of cystic fibrosis, but of pericarditis caused by pulmonary tuberculosis.
Our immediate response to the challenge of the pandemic outbreak was to launch a COVID-19 diagnostic laboratory, a legally licensed entity that provides diagnostic services based on our large staff of medical geneticists and diagnostic technologists.
As the only institute of the Polish Academy of Sciences, we have established a COVID-19 diagnostic laboratory with its own drive-through diagnostic point, which screens several hundred people a day. In addition to diagnostic tests that are performed, we analyze the SARS genome RNA structures, which are deposited in global databases. By tracking the pandemic dynamics, we have shown that the UK variant was responsible for the third transmission wave, representing 96% of cases. As the COVID incident peak can overlap with the influenza peak, we have developed, and as the first in Poland, introduced a PCR test to differentiate between SARS-CoV-2 and influenza virus infections.
Our pioneering work in the field of regenerative medicine was a response to a problem of high mortality rate caused by heart diseases worldwide. Addressing above issue as the second group in the world, we applied directly into a damaged heart myoblast, cell responsible for skeletal muscle regeneration. This procedure was reported by Reuters. Next, we focused on improving cells delivery and effectiveness of the therapy. This time, as the first in the world, we use endoscopic methods for percutaneous myoblast delivery. This procedure was associated with reduced risk and it shortened patients' recovery time.
Finally, in 2017, a team of Poznań cardiologists and geneticists performed pioneering delivery of genetically modified muscle cells to so-called non-option group without perspective of treatment. Further optimization attempts of cellular intervention to the heart are pursued.
We are using genetic cell and tissue engineering to investigate the mechanism of heart development and regeneration. We are working with induced pluripotent stem cell. Their exceptional features allow them to form any cell type found in our bodies, including heart cells. Combining iPS-derived heart cells with matrix and other cell types, we have established an engineered heart tissue model. Thanks to EHT unique features, we use it to evaluate biosafety of drugs and to model genetic heart diseases. In the future, the EHT could be used for fixing myocardial infarction scars.
Colorectal cancers are the most common in humans. Some of them are hereditary and develop on the basis of precancerous stages as multiple polyps of the large intestine. At the Institute, we have the largest collection of DNA samples in Poland from over 1,000 families with strong predispositions to occurrence of the disease. As the first in Poland, we have developed innovative methods of familial adenomatous polyposis molecular diagnostics and proposed treatment regimens. In studies of inflammatory bowel disease, including Leśnowski-Crohn disease, we identified the genetic basis of the severe course of the disease and identified new markers for personalized treatment.
At the birth of their child, the parents can hear son or daughter. However, it's not infrequent that sex assignment can be problematic because of intersexual characteristics. We work to provide more precise genetic diagnosis of those young patients that, as we believe, will facilitate decision-making about sex assignment as well as appropriate medical care. In turn, infertility is frequently caused by disruption of genes that are necessary for production of male and female germ cells. Given that the present diagnosis of infertility is not sufficient, we have established methods for detection of infertility-causing mutations. This means progress in diagnosis of infertile couples. We also investigate such mutations to understand how they disrupt processes in germ cells. This may help to provide new therapies of infertility in the future.
Our research on semen biology, conducted with the use of modern research techniques, has been the key factor in the dynamic development of andrology. We have been successfully identifying new non-invasive biomarkers of various types of male infertility caused by both genetic and environmental factors. Our research projects focus on examining a wide variety of unconventional semen parameters and aim to pinpoint the critical points in the process of male gamete production and maturity. The results of our pioneering molecular research are integrated with everyday medical practice and lead to the development of new diagnostic and treatment methods of male fertility disorders.
Leukemia is the most common cancer in children. We aim to get to know the mechanisms of this disease. We use advanced methods that allow the patient's leukemic cells to be tested for all genes simultaneously, for their abnormal structure and activity. We want to identify the features that will allow to personalize the treatment, which is to adjust the therapy to the specific features of leukemia in individual patients. We were the first in Poland to introduce studies monitoring the number of leukemic cells in patients treated with chemotherapy and after bone marrow transplantation. Over time, these tests have become the standard. They are now performed in all children with leukemia, which has increased the effectiveness of treatment.
So far, genetic studies have focused on genes from which proteins are produced.
But if we imagine our genome as a pizza, genes encoding proteins are only a tiny piece, about 2%. What about the rest? Until recently, it was considered as useless junk DNA. However, recent studies have shown that the non-coding DNA regulates genes and is crucial for proper functioning of organisms. We are the leader in Poland in the field of non-coding DNA studies in hematologic malignancies. We do not focus on the ingredients, which are genes, that make up the cancer cell, but on the cook, which is the non-coding DNA that determines their proportions. The results of our studies will enable development of novel, more effective and specific drugs.
Tu macie taki płyn. My tym płynem normalnie karmimy komórki, które hodujemy. Puść balonik i teraz spróbuj nakropić w te kropeczki. No to co, to teraz parę kropek żółtych zrobić, co?
We put much emphasis on activity for the benefit of society, both by applying the results of our research in practice and by popularizing science. The latter objective is supported by the activity of the geneticists association that we have established.
The participants have an opportunity to observe scientists' work. Moreover, we organize workshops in educational institutions as a part of the mobile school. We participate in all important events popularizing science, such as Researchers' Night. Our goal is to create social awareness concerning the importance of genetics in the modern world.
The DMD gene is the largest known human gene in which a huge number of mutations can take place, causing the disease, Duchenne muscular dystrophy. Previous therapies using precursor muscle stem cells have turned out to be ineffective, so we are making attempts to obtain mature muscle cells. Correcting their defective gene will allow them to regain functionality and aid muscle regeneration. The development of new therapies is of great importance for patients and their families.
The history of molecular research used for the purpose of forensics is over 3 decades old. The first genetic analysis of DNA fingerprints was conducted as a part of investigation into the plane crash in Kabaty Forest near Warsaw. This analysis was carried out at our institute, which was first time performed in our country to indicate that such analyses are possible, and we fully implemented DNA tests for identification purposes. Such tests are standard procedure nowadays.
The launch of the innovative medical center of the Institute has been an extension of our activities for the public. The center continues the existing diagnostic activities for the SARS-CoV-2 virus and also offers a wide range of tests for rare genetic diseases, cancer, and various infectious diseases. To the best of our knowledge, virtually every human disease has its genetic component. In the era of modern personalized medicine, based on scientific evidence and focused on treatment of the patient with consideration of his/her genetic uniqueness, our activities have unquestionable and ever-increasing social significance. We invite you to the Institute of Human Genetics of the Polish Academy of Sciences in Poznań.